Hi, everybody. Hi. Welcome to Hot Science Cool Talks. Congratulations for being here. This is quite an evening. I'm really looking forward to this. I want to thank all of our supporters up here. What an amazing group of people. Let's hear it for all of these folks who help support what we get to do for free. How about all those activities out there? Thank you, all you folks who put this on. How many of you people held a skull of something in your hand tonight? What? A lot of you did. And even more. Let's hear it for these folks who put on all the activities. Girl Scouts of Central Texas, are you here? Oh, you didn't make it. That's okay. Or you're here and you're a little bit shy. That's all right. How about the Bernice Hart Early College Prep Elementary School? All right. How about the UT Osher Lifelong Learning Institute? Yes. How about the UT Longhorn Band? Oh, I thought they were going to be... Please remember to silence your devices. No talking during the talk. Only Dr. Kirk will be talking during the talk, but he wants to hear all of your questions afterwards. And this brings us to the 127th Hot Science Cool Talk. If you started this program and that doesn't make you feel old, yes. And we're delighted tonight to have Professor Chris Kirk talk to us. He is a professor in the Department of Anthropology here at the University of Texas. And Chris was born and raised in Austin, Texas, and his household was very formative to what Chris wound up becoming. I want to tell you about how formative his household was right here, right now. When he was a kid and he had a question about something about how the world worked and he asked his parents about it, what they would do is they'd pull out an encyclopedia, because this is like pre-internet, and they'd look at diagrams, they'd draw pictures, and he became just a sense of wonderment about the world around him and how it worked. Then he went on to go to Westlake High School, and we'll get up to present day pretty soon here. That was very formative for him too. And then guess what? His parents were both Longhorns. They met in class. They left the university and joined the Peace Corps. Went all over the world. They came back. They had Chris. Chris became a Longhorn himself. So 3 generations worth of Longhorns we're going to hear tonight from Professor Kirk. He did his PhD at Duke University, where he spent many hours in the company of lemurs and lorises at the Duke Lemur Center, where he also met his wife in class, just like his parents met in class, at Duke University, and his wife, Rebecca Lewis, who is a former Hot Science Cool Talk presenter here as well. Just amazing. It's a very small world. And Professor Kirk, has participated in paleontological research all over the world, including Egypt, Botswana, Turkey, various sites across the United States, especially some stuff you'll hear about tonight in the Big Bend region of far west Texas. Right? He's an expert on primate evolution, particularly interested in using the relationship between form and function to better understand adaptations and ecology of various fossil species of primates. He's also a member of UT's elite Academy of Distinguished Teachers, which is like being a Top Gun flight instructor, right? And that his motto is that he is truly, quote, not in the business of good enough. And tonight, I think you'll see why. Please welcome Professor Kirk.
I have a question for you. Is anyone here afraid of the dark? If so, I want you to imagine that you're a katydid on the island of Borneo, a large, delicious katydid. Night has just fallen, and you're cleaning your antennae and looking forward to a nice long night of eating leaves in the tropical forest ahead of you. Isn't it great to be a katydid in Borneo? Don't you love it here? Hold on a second. Did you hear something? No, I don't think you could have heard anything. You must be imagining it. You're so well camouflaged, nobody could possibly find you at night in the forest. Wait, maybe you did hear something. Hold on, it's too late. That's right, and the terrible beast is upon you. You've just been eaten by a tarsier. That's right, insect slayer, nightmare of cicadas and katydids, tiny terror of the tropics. Okay, so for the record, I wanted to call this talk Talking Tarsiers, Tiny Tree-Dwelling Terrors of the Torrid Tropics, and Jay said, maybe you should shorten that down a little bit. I think that was probably a good idea. This handsome mug belongs to a tarsier. And do you want to see what I just described to you in action? Ready? Okay, good. Here we go. Uh-oh. Oh! I realized that was really fast. Let's see it in slow motion. So there it is. Okay, look. The terrible beast leaps in from off screen, gets into position, locks on target, and when the moment is right, it leaps. Here it comes. Oh, gruesome. Oh, the humanity. A bite to the head takes care of that insect. What a great animal. So, yes, meet the tarsier. And I like to say that tarsiers are one of the most interesting animals that most people have never heard of. I imagine that you all want to know something about tarsiers, and that's why you're here. So let's get started. First things first. Yes. Tarsiers are primates. Primates include monkeys and apes and humans, but they also include some types of mammals that people often leave off the list, like lemurs and lorises like this potto and bush babies, and there is a tarsier. Tarsiers are only found in one part of the world, and that is in a series of islands in Southeast Asia. And if we zoom in on this region and I add the range of living tarsiers, what we're looking at is a range that includes the southern Philippines, the southern part of the island of Sumatra, and then this big island here, Borneo. It's right on the equator. It's the world's third largest island. And then this amazingly shaped, let's go back. This amazingly shaped island right here, Sulawesi. And yet, well, as you've already seen, Sulawesi, I'll come back to that in a second. But let me say, what's amazing about tarsiers over this range of islands is there's not one species, there's not two species, there are 14 species of tarsiers across this range. And if you look closely, what you'll see is that all but two of these species are located on the island of Sulawesi and the small outlying islands. So it's pretty impressive. This as you travel around Sulawesi, every little peninsula that you go to, there's a different species of tarsier living there. And Sulawesi itself is in a really interesting place geographically. It's marooned between what geographers call Wallace's Line and Weber's Line. And to the west of Wallace's Line, you see the typical sorts of mammals that you find in Southeast Asia, like tigers and elephants and monkeys and rhinos. And to the east of Weber's Line, you find the typical sorts of mammals that you see in Australia and New Guinea today, like kangaroos and platypuses and cuscuses and other types of marsupials. Look, there's Sulawesi in between Wallace's and Weber's Line. And what's interesting is even when sea levels were lower than the present day, for example, just 12, 15,000 years ago during the last ice age, Sulawesi was still surrounded by deep water channels. And so you had to cross water to get to Sulawesi. Surely this is part of the explanation for why there are so many different types of tarsiers there. All right. So, yes, tarsiers are in fact small. Terrible though they may be to insects, they are tiny. They could perch on your head or on your outstretched finger. The name tarsier itself comes from the word tarsus. Tarsus is what anatomists call your ankle bones. And so what I've done in these pictures is if you look at the asterisks, those are actually on the heel of the tarsier's foot in each picture. So that means this whole thing is the foot here and here. They have incredibly long feet. And one of the reasons they have such long feet is there are two tarsal bones, ankle bones, that are extremely elongate in tarsiers. These are the calcaneus and navicular. And so you're just looking at a bunch of foot skeletons of different primates. And there's the calcaneus and navicular. And here I've labeled the same bones in humans so that you can see that they're normally short, chunky bones. But in tarsiers, they're so long and skinny they look like long bones. So they give tarsiers these really long feet. I'm not used to having Jay gave me this laser pointer that I can advance with. I keep running back to advance the slides in case you guys are wondering what I'm doing. Look at this. Let me put it all in perspective for you. If you take data for Bornean tarsiers, head and body length is about 150 millimeters. The length of the foot at 71 millimeters, that is almost half of head body length. Imagine if humans had feet that were half of our head and body length. Or instead of imagining, I just happen to have a picture of Professor McConaughey here. Class of 93, UT's Minister of Culture. And he is a typical human in having a foot that's about 30% of head and body length. So if we want to give him tarsier-sized feet, this is what it looks like. So I measured it. I did the maths on this. That is 47% of head body length that would look like he's running in clown shoes. These long feet go with long legs and thickly muscled calves. They look like speed skaters in the Olympics. And these are adaptations for leaping. Tarsiers are leapers par excellence. They live in tropical forests, usually relatively low and close to the ground within the bottom 2 meters of the forest. But in order to get from one arboreal support to the next, one little twig or branch, they leap from one to the next. And tarsiers, look at this. Tarsiers have been measured leaping more than 16 feet. 16 feet. This is a wee ickle little primate that can fit in the palm of your hand, and it can jump 16 feet with those frog legs. So that's 33 times head and body length. And if you want to put that in perspective, elite NCAA long jumpers, right? They can jump about 22 to 27 feet, and that is only 7 to 9 times head and body length. If humans could leap the same distance that tarsiers can relative to their body size, they could start on the nose of a 737. They could leap over the entire plane and clear the tail. Because the 737-500 is about 102 feet long, and you'd be able to leap about 100 feet if you could leap 33 times your head body length like a tarsier can. More amazing anatomy. Has anybody noticed that tarsiers have gigantic eyeballs? I think so. Yes, indeed. Good. There it is. So relative to head and body size, tarsiers do in fact have the largest eyeballs of any living mammal. Think about what that means. Pick anything with fur that makes milk for its babies, and that you can think of, and if it's not a tarsier, you're thinking of something that has a smaller eyeball than a tarsier relative to its head size. So this illustration, let's go back. This illustration helps make the point. We're looking down on the top of a tarsier cranium, tarsier skull, and the eyeballs are in place. And what I want you to notice is the eyeballs are so big, they hang way outside the eye sockets. Tarsiers actually have to have big, thick ligaments basically in their eyelids to hold their eyeballs in place because they hang outside of the eye socket. So a typical tarsier is going to have a head length of about 40 millimeters. It'll also have an eye length of about 18 millimeters. So the eyeball is 45% of head length. I finally do get to come over here. That's why tarsiers have skulls that look like this. And remember, the eyeballs are hanging outside of the orbit. Let me show you what a tarsier eyeball actually looks like. By the way, door prize later on. One of you is walking out of here with this. All right, so this is me some years ago. I wish I showed the full picture because you could see that I had hair back then. But I've got a tarsier eyeball in this hand, and I've got a tarsier cranium in this hand. And the first thing I want you to notice is that this gigundous eyeball goes in this tiny little head. But then I want you to notice this clear bit in the front of the eye, right? The darker part, that's the cornea. That's what lets light into the eye. Compare the size of the tarsier's cornea to my own cornea. It's bigger than my cornea in a head that size. So a human head length on average is about 200 millimeters. I slapped calipers on myself just to make sure this is true. Human eye length is about 24 millimeters. That's only 12% of head length. So if humans had eyes that were 45% of head length like a tarsier, that would be 90 millimeters long. 90 millimeters. People, a baseball is only 75 millimeters in diameter. This figure is closer to a softball. Imagine humans with softball-sized eyes. Or maybe instead of imagining it, It's amazing what you can find with Google image search. I just happen to have a picture of Professor McConaughey with a softball. And you can take that softball and compare it to the size of his face. You know, it's about the size from the bottom of his nose to the base of his chin, right, when he's smiling. So then we can take a better picture of Professor McConaughey, and we can tarsify him. So, and again, I did the math. That's what it would actually look like. I think we can all agree that it is good for him that he doesn't have eyes that look like this, if for no other reason than the fact that his students would surely call him Professor Peepers behind his back. More interesting things about tarsier ecology, their diet is 100% other animals. There are hardly any other primates out there that are like this, right? So mainly insects, the occasional bird, the occasional lizard or unlucky snake. This is what tarsiers eat. The vast majority of primates, if you offer them a piece of fruit, they'll be like, great, I love fruit. Tarsiers are like, no, I only want to eat other animals. So they have these sharp, pointy teeth and befanged mouths that you expect to see in a super predator like this that are well adapted for dispatching and slicing up insects. Look at that, amazing. No other primate has teeth like this. They've also got, perhaps you've noticed, very, very big hands and very long fingers. This is part of their basic predatory adaptation. But first, I just want you to see how big these hands and fingers are. We've got, from probably somebody purchased it in the 1940s, we have a tarsier skeleton in the anthropology teaching collection. And what I did is I just put the wrist on the back of the head, and I arched the hand and the fingers up over the top of the head. And so what I want you to do now, take the palm of the heel of your palm, put it on the back of your head like so. Now reach over the top of your head and scratch between your eyes on the top of your nose. Oh wait, you can't, because you're not a tarsier. By the way, I just happen to have a photo of Professor McConaughey showing his hands. And once again, this is to scale. There are tarsier-sized hands. So I think it's safe to say that if you had hands like this, he wouldn't need one of those foam hook'em hands to wear at the football games. Anyway, I mentioned that these hands are good for, they're part of their predatory adaptation. So what you see here, I love this photo that was posted online. Here's a tarsier, it's leaped, it's coming in, the feet are about to land on this stick. There obviously is an insect who's experiencing its last milliseconds on the planet. And look, the tarsier's eyes are locked on target, and the giant man hands are starting to unfold to ensnare the prey. And so here's the sequence that you've already seen, but let me break it down. Tarsier sees an insect, right? They're excellent leapers. They leap toward the insect, and at the last second, the hands open up like umbrellas. And even if the insect tries to jump away and evade, there's a good chance they'll still snatch it. Once they snatch it, it gets shoved into the mouth for the killing bite. Now, tarsiers used to think, tarsiers, scientists and tarsiers, the tarsier scientists, they used to think that all tarsiers were solitary. And then they actually went out and started observing tarsiers in the wild. And lo and behold, it turns out that, wow, they were wrong, as you could probably tell from this picture. It turns out, right, no surprise, there are 14 species. Some of them are indeed solitary. Males and females live alone and only come together to mate. But some of them are pair living, and they sing a little sweet, sweet duet together to mark their territory. Some of them are even group living, as you can tell from this big ball of tarsiers, wondering why somebody is taking a photo of them in the middle of the day. More interesting things about tarsiers. Tarsier females have singleton births. They have one baby at a time. That is really normal for primates. It's not at all normal for small mammals like this. Most small mammals are going to have multiple babies at a time. And when the mom wants to go out from wherever they've been sleeping and then go hunt, what she'll do is she'll carry the young baby in her mouth, and then she will park it somewhere while she's hunting, in the crook of a tree, a tangle of vines, or something like that, and the kid just sits there waiting while mom is off hunting insects. More interesting things about tarsiers. Like all primates, infants take a long time to mature. I looked up these data just a few hours ago. I realized I was missing this from the talk. Gestation, the time that the baby is in utero growing, 6 months. Again, a tiny, tiny furry little mammal. Your typical rat that's about the same size, 3 weeks of gestation versus 6 months of gestation. And this is the typical primate strategy. Have fewer babies and invest more in them. Take good care of them once they come out. And then weaning doesn't happen for 80 days. Your typical rat is weaned 3 weeks after it's born. So the kid is still there, 80 days later. Mom, I'm hungry, I need some milk. Anyway, this is not at all unusual for primates. This is very unusual for mammals in general. Well, as you can imagine, from all of that anatomy that you just saw, when tarsiers were first discovered, nobody had even the remotest idea what to do with them, how to classify them. One of the first accounts in the published literature comes from this guy, Georg Kamel, who was one of the first European naturalists in the Philippines. And in 1706, he publishes a letter and he says, hey, I'm paraphrasing, hey, there's this weird animal with giant eyeballs that's only active at night. I think maybe it's some kind of monkey. And then people started getting tarsier specimens pickled and shipped back to Europe. And so taxonomists like Christian Erxleben here took a look and he said, I don't think it's a monkey, maybe it's some kind of lemur. But we know that lemurs are only found in the highland of Madagascar, so it can't be a lemur. And so I love what this guy, the Count of Buffon, did. He was a French aristocrat and naturalist who published a very popular encyclopedia of natural history. And in 1765, he says, hey, I know what to call them. Let's call them les Tarsiers. That's what they are. And I have this great quote from Buffon. He writes, we've obtained this animal by chance and from a person who could tell us neither where it came from nor what it was called. So he's like, yeah, this amazing creature. I have no idea anything about its background. But it is very remarkable for the excessive length of its hind legs. It is from this very noticeable characteristic that we have derived its name. So when you say the word tarsier, you can thank Buffon in the 1700s for naming it a tarsier. Sadly, Buffon was even more wrong about what it was. He said, maybe le tarsier is some kind of jerboa. Qu'est-ce que c'est, jerboa? Anyway, jerboas are rodents. It's not a rodent, it's a primate. Buffon, right? I mean, come on, there was a lot that scientists didn't know at this time. At least he knew that jerboas have incredibly long feet, just like tarsiers. But unlike tarsiers, look at this. Look at these tiny T-Rex arms. Definitely not a tarsier. In fact, it wasn't until 1918 that this guy, Reginald Peacock, who is a British zoologist, he said, and again, I'm paraphrasing, he basically says, hey, has anybody ever noticed that tarsiers have dry noses? You guys know about dry noses and wet noses in mammals, right? How does your cat wake you up in the morning when it wants you to wake up and it's like, stop sleeping? You get the wet nose treatment? It turns out that some primates, like this lemur here, this type of lemur called a shafaka, they resemble lots of other mammals, most other mammals in fact, in having a wet, hairless nose, a juicy nose even, if you will, like a dog, like a cat. You've all seen it before in your own home, right? Anatomists call this a rhinarium. So a wet, hairless nose is a rhinarium. It's right there, a dime a dozen among mammals. Additionally, something that often goes along with a rhinarium is a split upper lip. So there's the split upper lip, there it is in the dog, you've seen it in a cat. This is a collecting surface. You know what dogs do with their rhinaria? They stick them on things that they want to smell, like other dogs' parts, and you're like, hey, come on, Fido, what are you doing? Well, tarsiers are like monkeys, apes, and humans in being really unusual for mammals. I'm going to keep using these terms, superlative terms like bizarre and unusual. They don't have a rhinarium, right? Most mammals have rhinaria. And tarsiers are like monkeys and apes and us in having a dry nose. They don't have a split upper lip. Look at that nice hairy lip, just like a monkey. So lacking a rhinarium, it's pretty unusual. This is great because Peacock was really the first scientist to recognize that tarsiers are actually our close evolutionary relatives. And let me tell you what I mean by that. So don't be afraid of the diagram. I'll walk you through it. So these sorts of diagrams, everybody's seen a family tree, right? So that's how you represent family relationships. This is like a family tree. It's just showing evolutionary relationships. So you got all the major groups of living primates here, lemurs and lorises and bush babies here, tarsiers in the middle, monkeys, apes, and humans over here. And what I tell my undergrads is these diagrams are easy to read. Every place you see a branch point, think of that as a common ancestor at some point in the past. So you have a lineage evolving, and then look, there's a split point. One branch goes to lemurs and lorises, one branch goes to tarsiers, monkeys, apes, and humans. That split point represents the last common ancestor of all living primates. And then we follow this branch further, and here's another split point. One branch goes to tarsiers, one goes to monkeys, apes, and humans. I've highlighted it in red because that represents the last common ancestor of tarsiers, monkeys, apes, and humans. Hakkak named this group Haplorhini. Haplorhine just means simple-nosed. So the haplorhine primates are the simple-nosed primates that don't have a rhinarium. And now we know from molecular data that this is indeed how tarsiers are related to other types of primates. Okay, so you heard in the introduction that I study fossils. You could be forgiven for wondering, is this guy just some weird obsessive tarsier fanatic? Maybe guilty. But I will say, right, if you're wondering, why is this guy who studies fossil primates so into tarsiers? I have several answers for you. One is that the fossil record of definitive tarsiers is really interesting, and it goes all the way back to the Eocene epoch. The Eocene is this period of time between about 56 and 34 million years ago. So we mean ancient, right? Long before humans have evolved. And what you're looking at here is a picture of some paleontologists collecting fossiliferous sediments at a site in what is today China called Shangwang. It's just west of Shanghai. And what it is, you can even see the color change here. See how these sediments are more brown and these are different? This is a filled-in cave site. So caves have a life. They open up as groundwater, which is slightly acidic, eats away at limestone, and then they're open for a while, and then they fill up with stuff. And carnivores den in caves, and things wash into caves. So what these paleontologists are collecting at this site of Shangwang is a bunch of 45 million year old small mammal fossils. And these are mostly small mammals that had the misfortune of being eaten by owls. So look at this. So just as in the tropics today, in the tropics of what would later become China, so 45 million years ago, owls are flapping around the forest at night, and they are espying tasty, juicy little rodents and other things. And they'll snatch them, and then what they do, right? They don't have teeth, they don't have cutlery, they just down the hatch, the whole animal. And then they digest all of the soft, tasty bits. And then when they're done, what comes up is this. It's a wad of the undigestible bits, the fur and the bones and the teeth. So 45 million years ago, you've got owls denning in caves, and every night, bark, bark, right? They're basically barfing up owl pellets, leaving a record of what they ate 45 million years ago. Guess what was on the menu? Tarsiers. One super predator eats another. There you go. Look at this. Here are the tiny teeth of a fossil tarsier from Shangwang, 45 million years old. And I'll tell you right now, you don't spend all day looking at tiny teeth like I do, but trust me on this, these are the spit-in image of tarsier teeth. They are, and look, here is even, you don't believe that? Here is even a little fragment of a cranium that looks pretty much exactly like a modern tarsier. So similar to modern tarsiers that when this species was described, 45 million years old, the discoverers called it Tarsius eocinus, the Eocene tarsier. And the reason this is amazing is Tarsius is a genus that is alive today. Alive today. So, look at this. So here we got Renee Zellweger. What's her species? Homo sapiens. Our genus, right? Yeah, class of 92. Don't worry, all the famous, former longhorns that I can find, they'll be included. In any case, genus Homo has been in existence for less than 3 million years. So we've got evidence from the fossil record of East and Southeast Asia that tarsiers have been around acting like tarsiers and doing tarsier-like things, snatching bugs at night and leaping around tropical forests for 45 million years. That is a long time, people. A very long time. And I know some paleontologists hate this term. I hope nobody's cringing. Tarsiers really are sort of living fossils, right? Their ecology, as far as we can tell, has been the way it is for a long time. So anyway, most of my fossil research is in North America. And you don't find Tarsius eocinus in North America, but you do find a really interesting group of Eocene fossil primates called omomyoids. And so this is a view from one of my study sites. This is at the Dalquest Desert Research Site out in the Big Bend region of Texas. And I like to show this picture because this tells the true story for paleontology. I like to say, here's the glamorous business of field paleontology. Sitting around on your tuchus, ooching forwards, looking for tiny jaws and teeth. And if you are super, super lucky, you find something like this. So there is an omomyoid partial mandible. And let me show you what slightly more intact omomyoids look like. So this is a pretty spectacular fossil that is 51 million years old. It's from Wyoming, and it's a genus called Shoshonius. And what you're looking at here is a cranium, right? Most of the skull in side view. There's the nose, there's the upper jaw and teeth. Here is the eye socket, right? Does it take a trained anatomist to see that that's gigantic? It does not. Huge eye sockets, almost certainly nocturnal like a tarsier. Cheekbone, there's the brain case. So let's add it all up. Shoshonius is a good example of an early Eocene omomyoid. And they're in North America, they're in Europe, they're in Asia. And so what we see is that they start out really small. So Shoshonius is about 115 grams. That's in the tarsier size range, check. They have huge eye sockets, as you've already seen. Sounds like a tarsier, right? Check. And their diet, if you look at their teeth, it doesn't look like they are mostly as specialized for only eating insects the way tarsiers are. They probably ate some fruit, but it's really clear that almost all of these omomyoids had insects front and center in their diet. Check. So you won't be surprised to hear that for most of the time that paleontologists have been finding these omomyoids, they have been, rightly wondering if these are early relatives of tarsiers. So in other words, at the same time you have proper fossil tarsiers in East Asia, you have possible early tarsier relatives in North America. It's not that far-fetched given what we know about the fossil record. One thing I will say about Shoshonius, though, is that it did not have the huge hands and feet of a tarsier. But you can tell from this artist reconstruction what they were thinking of when they reconstructed Shoshonius. Look at this rat tail with the tuft on the end. That's a tarsier tail. By the way, there are omomyoids from the Eocene of West Texas where I work. So this is a view right along the Rio Grande in far western Presidio County. It's an area called Rifle Range Hollow. And I was just out there in May, fortunately. And there's a little cairn right over here where the arrow is marking the site of the discovery of this unbelievable fossil. This was found in 1965 by this man, Professor John Andrew Wilson, who was a professor of geosciences here at UT and who founded the Vertebrate Paleontology Lab, which is still the nerve center of all fossil work here at UT. Runia is still to this day one of the most complete and certainly the least distorted cranium of any omomyoid found anywhere in the world. I like to tell visitors, I know people love dinos and Quetzalcoatlus and things like that. I like to tell visitors that it's really one of the gems, the jewels of the collection of the Jackson School Museum of Earth History, their paleo collection. Anyway, it's 38 million years old. It's an omomyoid. There were really interesting omomyoids in North America, including where I work out in West Texas. So I've been collecting Eocene fossil mammals really since about 2004. And a lot of the primates that I find are these omomyoids, like Runia, like Shoshonius. And so this is just a view to show you one of my study areas in sort of north central Big Bend National Park. This is just north of Panther Junction, if you've ever been out there. And let me say this before I go any further. The Big Bend in the Eocene was a very different place than it is today. Don't think desert, think moist, humid, tropical forest, probably with a closed canopy of trees. And then don't think of sort of jagged, well-eroded highlands like the Chizos Mountains or the Davis Mountains. Think active volcanoes off in the distance, supplying the sediments that are filling in these basins. And so here is an artist's reconstruction of one of my study areas 44 million years ago in the Eocene. Volcanic highlands with active volcanoes, tropical forests, and a couple of different species of omomyoids there. And one of the things, I don't want to spend too much time on this, but I have to tell you, one of the things that makes studying omomyoids in Texas really interesting to me is most of the species that we find in this part of West Texas, they're not found anywhere else in the world. They're unique to this area in the Eocene, as far as we can tell. So here, for example, are jaws and teeth of a genus and species that a co-author and I described in 2008. We called it Diablomomys dahlquesti. That's this guy right here. Here's a new species that my co-authors and I just described a few weeks ago in Journal of Human Evolution. We called it Mytonius williamsii. And right, they're both only known from the Big Bend region of Texas. So if you're a paleontologist and you're interested in fossil primates like me, and you're finding omomyoids, you have lots of questions about them. And front and center for me is now, and has always been, how are omomyoids related to living primates? I know that I've already told you that a lot of people think that they're related to tarsiers, but let me just remind you, these diagrams are just showing you the evolutionary relationships the same way a family tree shows you family relationships. So we know that omomyoids are primates, and the question is, which branch do they link onto? Are they more like lemurs? Are they more like monkeys? Where do we slot them in? And I would just summarize a lot of literature and say there are two main ideas for how you fit omomyoids into the living primate tree of life. And if there's a majority point of view, it's this one right here, that omomyoids are more closely related to living tarsiers than any other kind of primate. So there's that split point. That's a common ancestor for omomyoids and tarsiers, exclusive of all other primates. There's also a minority point of view that, well, maybe they're not primitive tarsiers, maybe they're early haplorhines. Remember, this group is called haplorhini, the simple-nosed primates. Maybe they branch off of that lineage leading to haplorhines before the time of the last common ancestor of haplorhines. So, do you want to see some evidence? I figured I had to show you some of my own research. I couldn't just show you tarsier videos all night. Although if I took a poll, you might actually vote for tarsier videos. I realize that. Let's consider some evidence. I have a question. Have any of you ever looked into the nose of a cow skull? Raise your hand proudly if you have. Yes. Curious people. Good. So this is a cow skull that I have in my office. And if you've peeped into a cow skull, this is what you've seen. So you have seen paper-thin scrolls of bone hanging out in the nasal cavity that are called turbinals. Turbinals. And in life, turbinals are covered with a mucous membrane. That shouldn't be surprising. Everybody knows it's juicy inside your nose, right? All right, so these paper-thin scrolls of bone are covered with a mucous membrane. There are two kinds of turbinals in there. Type 1 are basically used for air conditioning. These are called respiratory turbinals. And what they do is they warm and humidify air as you're breathing in through your nose. Now, I'm not interested in respiratory turbinals as much as I'm interested in the turbinals that are used for smelling stuff. Everybody knows that you smell with your nose. Inside your nose, you have another specialized type of mucous membrane that sits on a special type of turbinal called an olfactory turbinal. And that's the site where you will actually detect odors inside your nose. So if you can say something about the anatomy of the turbinals, you can say something about the sense of smell. And that's great because turbinals are made of bone and they might fossilize under the right conditions. Let me just say this before I go on. The very best way to study turbinals, right? Turbinal anatomy understanding has been revolutionized through the use of 3D X-ray scanning or micro-CT. And I just want to say one of the great things that we have here at UT is the premier micro-CT scanning facility in the world. I hope you all saw the table earlier. Raise your hands proud. Yes, UTCT right here. I love you guys. Thank you. So, yes, and so what we see here is, look, it's like a crocodile skull being scanned, and someone's going to look at the internal anatomy of that skull. Pretty cool. So what I'm going to show you here is I'm going to show you one of these micro-CT scans, just 3D X-ray reconstruction of the skull of a typical mammal, in this case an opossum, and I'm going to do some violence to it digitally. I'm going to chop its nose off for you. Are you ready? I'm going to chop in this plane. Are you ready? 1, 2, 3, chop. I chopped its face off and its nose, and what I've done, you see these little curly cues in here? I have exposed the incredibly delicate and complicated olfactory turbinals. Remember, in life, they're covered with a specialized mucous membrane that lets you smell stuff. And the reason they're thrown into these folds and coils is to increase their surface area so you have more surface area inside your nose for smelling stuff. Does that make sense? Okay, good. So, let me break it down a little bit more. Things that are smelly are shedding chemicals into the air, right? Willy-nilly. They're dissolving in the air and floating around. And your job, if you want to smell them, is to somehow get those chemicals into your nose where they can physically bind with specialized receptors on that mucous membrane that covers the olfactory turbinals. That's how you smell stuff. Is everybody with me? Okay, good. So the general rule is this, right? If you've got more olfactory terminals, that will give you more surface area inside your nose for smelling stuff, right? And once you have more area in your nose to physically interact with those chemicals and detect them, you've got a better sense of smell. And so look at this. Here's Wes Anderson, class of 91. He is like, presumably, a typical human in having about 10 square centimeters of this mucous membrane for smelling things inside his nasal cavity. He's only got 2 olfactory terminals. I'll tell you now, it's pretty sad. Okay, look at this. And it's not Wes Anderson, it's all of us, okay? Check out this dog. 16 times the surface area inside its nose because of all of those complexly folded terminals, 12 to 16 of them. Who has the better sense of smell? The dog. Humans have got a particularly lousy sense of smell for a mammal. So, you could probably imagine this is where I was going. Micro CT images inside the noses, the nasal cavities of different types of primates. We got a tarsier up here, we got a monkey here, we got a lemur here. This is basically the palate, the upper teeth. There's part of the brain case, and these structures that have been colored in, right, in bright colors, those are the terminals. And I want you to, first off, ignore the red one, because the red one, that's just a respiratory terminal. I want you to pay attention to the orange, green, and blue terminals, the ones that are used for smelling stuff. And I've omitted some of the smaller terminals, but you can already see the story I'm going to tell here, right? Which one's got the better sense of smell? The lemur does, exactly right. Good, you were listening. Excellent. So, lemurs are typical mammals. They have lots of olfactory terminals. They have an excellent sense of smell. 7 to 8 olfactory terminals, in fact. I've left out some of the other ones that are harder to see in this view. Who are the sad mammals with the poor sense of smell? It's us and our close relatives, the haplorhines, the simple-nosed primates. It's hard to get more simple than only 2 olfactory terminals. There it is in a tarsier. There it is in a monkey. This is extraordinarily unusual. So now what am I going to do with fossils? I'm going to micro CT scan them at UTCT. And this is work, by the way, all science is collaborative these days. This is work that I did with Dr. Ingrid Lundin, who's now at Hunter College in New York. These are micro CT scans through the cranium of Shoshonius that you saw earlier. And what Ingrid has done is she's colored in all the terminals. Can you guess where they're going to fall? There they are. 6 terminals and counting. I only say 6 plus because it looks like some terminals just weren't preserved. So the point is, Shoshonius, an omomyoid, inside its nose, it does not look like a tarsier, a monkey, a haplorhine. It looks like a lemur. And remember, in looking like a lemur, it just sort of looks like a plain vanilla mammal with a great sense of smell. It's these guys. Oops, let's go back. It's these guys, the haplorhines, that are weird here. All right, you want to see some more evidence? Okay, good. Hope you haven't had too much. Tarsiers, monkeys, apes, and humans have a visual superpower that no other mammals have. And I'm going to illustrate this by showing you the inside of my eyeball. You are welcome. So here it is. You are looking at a view of my retina. You can see the lower eyelashes here. You can see some of the blood vessels that spread out across the inner surface of the retina. The retina is just the layer of cells in the very back of your eye that actually detects light. So all the light-sensitive cells in your eye, they are in the retina. And remember, I'm a haplorhine, you're a haplorhine, tarsiers are haplorhines, and we haplorhines have got this crazy thing. It looks like a pit, maybe a dimple. It's called a retinal fovea. Guess what fovea means in Latin? Pit. Yeah, the anatomists are like, what are we going to call this pit in the eye? Let's call it pit, but let's use the Latin name so it sounds fancy. All right, there it is. Here's my fovea in one eye. Haplorhines are the only mammals that have phobias, the only mammals with phobias. And what's amazing about these phobias is inside the fovea, you have this incredibly dense concentration of visual cells called cones. Cones are, right, these are some of the cells in your retina that are actually detecting light. And what you do when you look at something and you point your eyes at that item of interest, you are pointing the fovea in each eyeball at that item of interest. And this incredibly dense concentration of cones will sample whatever it is you're looking at and give you exceptional vision for details. Truly extraordinary. This is called having very, very high visual acuity. Visual acuity is just detail vision. So, Guess what haplorhines have in terms of visual acuity compared to other mammals? The highest known for mammals. Why? Because we have a fovea. Is everybody with me? Okay, good. I know it's a lot of anatomy. Look at this. Here it is again. Tarsiers are extremely weird. Tell you something you didn't know already. Look at this. Tarsiers are nocturnal, but they still have a fovea, and the fovea contains a high density of cone cells. Why is this crazy? If you learn anything about the retina and how it works, you know, oh, cones, they work in high light levels. There's this other type of visual cell called a rod that works in low light levels. So what tarsiers are doing is they're preferentially coming out at night when the moon is full and it's bright, right? That's when they like to be active. And they have these giant eyes for gathering in enough light that they can have cones functioning at night. And they got a fovea, and they've got the high acuity that goes along with it. Let me break that down for you in a slightly different way. Okay, you know that when you go to the eye doctor and they have you read lines off an eye chart, you're like E, F, P, T, O, Z. The more lines you can read and the smaller the size of the letters you can read off, the better your detail vision is, right? The better your visual acuity. So tarsiers have almost weirdly high visual acuity for a nocturnal mammal. Most nocturnal mammals don't rely on vision to the extent that tarsiers do. And so it would work like this. Take a tarsier and take an elephant. Take them into the eye doctor's office and have them read lines off of an eye chart. Guess who could read more lines? The gentle tarsier. And that's even though the tarsier has an eyeball that's 1.8 centimeters long. The elephant has an eyeball that's 4 centimeters long, the size of a ping pong. Each elephant's eyeball is bigger than the tarsier's entire head. But the tarsier has better detail vision because it has a fovea that's crammed full of cones. Isn't that amazing? I think it's amazing. I know what you think. Okay, hold on, wait, guy. He was talking about fossils, and now he's talking about tarsiers in an eye doctor's office, and they can't read, so that wouldn't work anyway. Listen, you can't measure visual acuity in a fossil, right? You can't take an omomyoid like necrolemur from the Eocene of France. Guess what necrolemur means? Dead lemur. I love that genus name. You can't have them read lines off of an eye chart, right? But listen, if you think about the way retinas work, retinas do not exist in isolation. The cells in the retina at some point have to tell your brain what they have seen, right? And trust me, this is the least gross picture of an eyeball that I could find, so, all right. Anyway, look, here's a pig's eye. There's a retina in there. The retina communicates with the brain via this big, thick nerve called the optic nerve. And it's just filled with nerve fibers that are sending information from cells in the retina to the brain. So, tarsiers and other haplorhines have got a huge optic nerve. Huge even, with a Y, huge. So they got a huge optic nerve, and that's because they have a fovea with a ton of visual cells that need to send high resolution information onto the brain. So this is how it's going to work. You can't have necrolemur read an eye chart, but you can measure the size of the hole in the back of the eye socket that the optic nerve of necrolemur passed through, right? And if it's big, that's like a haplorhine. That's like a tarsier, a monkey, or an ape and human. If it's small, it's like a run-of-the-mill mammal. Are you with me? I know what you're thinking. Some of you are like, hold on now. You have to really establish what we're talking about here. Look, this is just to show you for a bunch of primate species that as the size of the optic nerve gets bigger, the size of the hole that it passes through, the optic foramen, also gets bigger. So check. Additionally, you may think that, well, optic nerves are apples and oranges across species. Look at this. Here's 25 mammal species. This is the cross-sectional area of the optic nerve increasing on this axis, and the actual count of total optic fibers increasing as you move up this axis. So as animals have bigger and bigger optic nerves, they are sending more information to the brain. So now we can look at an osteological, a bony proxy for visual acuity and whether there is a fovea. And you guys are like, oh no, another bivariate plot. Trust me, this is cool. All right, look. This is just skull length increasing on this axis, so small heads down here, larger heads, largest heads over here. On the y-axis, this is optic foramen area, the hole that the optic nerve goes through. Small optic foramina down here, increasing bigger and bigger and bigger. Every single point that you see is one of 98 different primate species. I measured them, and I have color-coded them for you. In red are all of the non-haplorhine primates, lemurs, lorises, bush babies, and things like that. And then, in blue and purple, are the haplorhine primates, tarsiers, monkeys, apes, humans with a simple nose. There are tarsiers as purple triangles if you're not in the cheap seats in the back. So what this is showing you is, as expected, if you have a fovea, you've got a bigger optic foramen than primates that don't have a fovea. Show of hands, how many people think that omomyoids are going to have like big optic foramina and they have foveas? Anyone? Okay, a few. How many of you think they're going to have small optic foramina? Most of you are wisely deciding not to comment, I guess. That's fine. There they are. Yeah, so they got small optic foramina for their head size, things like Runia. They did not have a retinal fovea. They've got lower visual acuity. They don't look like tarsiers and they don't look like other haplorhines. And then last but not least, this will go really fast, but I have to show you, I'm very excited about this. This comes courtesy of Ben Rodwell. Raise your hand, Ben Rodwell. Thank you, right over here. And you may have seen him at a table earlier. Yes. PhD student here in anthropology at UT. Here he is presenting what I'm about to show you at the Society of Vertebrate Paleontology meeting, 2 weeks ago in Cincinnati. And I said, may I please show my hot science talk what you found? And he said, yes. Okay. So tarsiers have an amazing ability to, here it is again. They have an amazing ability to rotate their head. Look, the tarsier is looking away from you over its left shoulder, and it rotates its head more than 180 degrees. Here, I'll go back and forth. I should have made a GIF about this. And so nobody, as far as we can tell, has ever actually quantified this, but everybody who studied tarsiers is like, look, they're like owls. They can rotate their heads all the way around. They have this amazing rotational ability of their head on their neck. That's probably because their eyeballs are so big, it's hard to move them in their sockets, so they just move their heads anytime they want to look at something. So, and this is one of the great things about micro-CT scanning. What did Forrest Gump say? You never know what you're going to get. You never know what you're going to get. Because here's the fossil, and you can see, right, this is a skull of Shoshonius. There's some bones, obviously. A lot of what you're looking at is actually the adhering matrix. Matrix is what paleontologists call just the sediments that are still stuck to the fossil. And so, and Ben did this, if you make the sediments clear and you color code the bones, this is what you see. Everything in purple is the cranium of Shoshonius. Everything in green is some other kind of bone of Shoshonius. And this includes, among many other things, the very first neck vertebrae of omomyoids that have ever been discovered. So Ben's working this up for his dissertation right now. And again, I don't want to go into too much detail, but I just want to show you what these neck vertebrae look like, because again, it bears on tarsier relationships to living primates. This is the second cervical vertebra, the second neck vertebra in Shoshonius. It's C2, or the atlas, or sorry, the axis is what I meant to say. And this is one of the key vertebrae involved in head rotations on the neck. And all I have to do, right, one of the things that I love about what I do is if somebody explains it to you and does a good job, you don't have to be a trained anatomist. Look at this. Here is the same vertebra in a dwarf lemur, which has sort of a typical run-of-the-mill range of head motions on the neck. And here it is in a tarsier. Which one of these does not look like the other? It's the tarsier, right? Without going into details, what Ben's analysis is showing is that Shoshonius, right, the first omomyoid where we've been able to look at neck anatomy, it basically just looks like a plain vanilla small primate. It does not have any of the adaptations that tarsiers have for very high head and neck mobility. And that's in spite of the fact that, remember, Shoshonius has got gigantic orbits and presumably had gigantic eyes. It couldn't turn its head all the way around like an owl. So anyway, all of these lines of anatomical evidence are favoring this relationship. So in other words, here are omomyoids branching off the lineage leading to modern haplorhines, the simple-nosed primates, before the time of their last common ancestor. And if you're like me, when you first see this, you think, gosh, that's kind of too bad. Because it would have been great if there were things that really looked like tarsiers kicking around. You know, there were close relatives of tarsiers kicking around in these Eocene forests in West Texas and other parts of North America. But I want to reframe this for you and get you thinking about it in a different way. Because look at this. This evolutionary lineage is equally ancestral to all of these groups. This includes your direct ancestors. And if we've sampled animals that branch off of this lineage, they're telling us what those ancestors, distant ancestors of tarsiers, yes, but humans too, what it's telling us is that we have big-eyed, right, nocturnal bug eaters in our ancestry, right? I don't know when, go back maybe 63 million years ago, and you've got things that look like this in your family tree. So anyway, if I can conclude with a few take-home messages. Take-home message number 1, if you're an insect, consider somewhere other than Sulawesi for a nice tropical vacation. It's the place that's lousy with tarsiers, apparently. Number 2, as you've already heard, tarsiers, oh goodness, tarsiers are superlative in nearly every aspect of their anatomy that I mentioned. Number 3, tarsiers have been around for a long, long time, at least 45 million years, which is incredible. Oomamyoids probably were not fossil tarsier relatives specifically, but that's okay, because what they do is they shed light on the earliest stages of haplorhine evolution, and we're haplorhines, and that's pretty cool. So, last but not least, let me just say, I mentioned it before, science is collaborative. I have to thank everybody. ESI, thank you, Jay, thank you, Angelina. I'm so happy to be back for a 4th talk. Thanks to the sharp-eyed field crews who find all the fossils for me. I ruined my eyes in grad school. It's always an undergrad going, hey, what's this jaw? And I'm like, Eureka, we party tonight. Anyway, all the people who have allowed access and all the collaborators, and thank you all for being here. I really appreciate it.
Professor Kirk will answer questions, and then hold on to your ticket, because we're going to give away some great Tarsierish associated prizes, including a very unique skull. That is a softball. All right, who has a question for Professor Kirk? Yes, we'll come right here.
I just want to know how you stand at the top of one of those hills and decide where you're going to dig.
Oh, that's a great question. So I like to tell people that doing paleontology is the closest thing to being a scientist and still being able to go to Vegas, right? Because sometimes you'll read the old, journal articles or old field notes of someone. They'll say, there are lots of fossils here. And you'll do the geological background work and you'll say, this looks great. There are sedimentary rocks of the right age. And you'll take a trained field crew there and you'll look and there'll be nothing. So sometimes you get skunked. But at this point, at least in North America, it's really rare to come across a site that no one has ever collected before. You find them occasionally, and it's a big day when that happens. But you're usually following in the footsteps of others. They say in science you stand on the shoulders of giants. Jack Wilson, who I mentioned earlier, is one of the giants whose shoulders I stand on. I cannot go to a place in the Big Bend region where he and his students were not there before me. So you basically have to give respect to those who were there already. Does that answer your question? Excellent. Is she here?
Thanks for the lecture. It was really informative and enjoyable. But I was wondering about the selective pressures, if any, that tarsiers experienced because of their habitat and why they evolved in the way that they did.
Oh, man. Well, it's such a good question. I almost don't even know where to start. Selection for leaping, right? This is clearly a good way to get around the understory of the Southeast Asian rainforest. There are not that many other mammals that do this. And I didn't mention it, but there's a special mode of locomotion getting around the world called vertical clinging and leaping, where your body posture is vertical and you cling with your hands and feet. And when you want to go from one arboreal support or one tree or bush to another, you don't go down to the ground, you don't run along a branch like a squirrel. What you do is you just leap from one to the next. There are some other primates that have evolved vertical clinging and leaping. So Shapaka in Madagascar, type of lemur, Indri, things like that. Some types of small bush babies have got long feet like tarsiers and can leap. So what were the exact selective factors that led to that kind of anatomy and not squirrel-like anatomy? Squirrels do great, right? There are hundreds of species of squirrels out there. They do great in the trees. They can leap in the trees. They have not evolved tarsier-like anatomy. I honestly don't have an answer to your question, but this is sort of like a PhD level question. If you want to do a PhD, let me know. In terms of what selective factors lead to these big eyes, I think if you think about it this way, if you're active at night, you can do two things. You can either invest in vision, as tarsiers have, or maybe more sensibly, you can invest in other senses like smell and touch. If you're bats, you can invest in echolocation and hearing. There's something about tarsier ecology that favors still being able to see details at night. And so they do something that almost no other nocturnal mammals do. My guess is it's probably a combination of leaping and hunting insects and needing to reach out with a hand and basically hit the insect every time. Additionally, if you think about how you get from one arboreal support to another once you're a leaper, you can't smell your way across. You can't hear your way across unless you're an echolocating bat. You have to see the target. And so I think you asked about selection pressures. I think the vertical clinging and leaping, as well as the predation, is going to favor a lot of these visual adaptations. Good questions. Yeah?
All right, Chris, you're back here.
Yes?
Wait, what happens if the animal, like when it gets pregnant and it goes hunting for insects, what if it forgets about the baby?
So, sorry, what happens to the baby when
The question is, when a tarsier is hunting and it puts down the baby, what if it forgets where it left the baby?
You know what? I've never heard any stories of mother tarsiers forgetting where their kids are. But what I can tell you, let's say that the kid is not just going to stay in one place. Let's say it's gotten a little bit older and it decides to crawl over here while the mom's hunting. And the mom comes back and she's like, where's the kid? Guess what most primates do in that circumstance? They give out a lost call. So Jay mentioned my wife, Becca Lewis, here in the front row, who's a primatologist. She studies a leaping type of lemur in Madagascar called a shafaka. When a shafaka is lost and it wants to know where the rest of its group is, it gives a plaintive lost call that sounds like this. It goes, "Brr, brr." And everybody else hears them and they're like, oh, they're there. And they go, brr, fuck, brr, fuck, brr, fuck. And they leap back and they're reunited. I am certain that there is some sort of a lost call that tarsiers can use in that situation.
Chris, we're going to take a whole bunch of other questions, but instead I asked people, I said, could you just make some lemur calls for the rest of the QB?
Oh, okay. You want to hear my favorite lemur call? This is an injury. They go, rah, 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 rah. That's an injury. Thank you. Is Aaron Sandell here? I know he was tabling earlier. You guys want to hear chimpanzee? This is a pantoot.
Wow.
My throat kind of cracked. Sorry, that wasn't very good.
Now can you do Christopher Walken?
Are tarsiers related to eyes?
In the sense that eyes are primates and tarsiers are primates, yes, they are related. And they have a bunch of similar anatomical features that tell you that they're primates. Both tarsiers and eyes have forward-facing eyes. That's a primate characteristic. They have grasping hands and feet with pudgy little pads on the tips of their digits and nails instead of claws. They have long periods of infant dependency and development. So yeah, they are related to eyes as primates. However, eyes are part of that other group that branched off, the non-haplorhine group that includes lemurs, lorises, and bush babies. It's called strepsirrhine primates, which literally means the twisty-nosed primates, because they have a rhinarium. So eyes are strepsirrhines with a rhinarium. Tarsiers are haplorhines without a rhinarium. Sure.
Could you explain how they evolved to become entirely carnivorous and no longer omnivorous, I presume?
I don't know. I mean, it's so strange. It's almost unprecedented. I don't know why you would do that. It's so rare. I mentioned there's only one other type of primate that's like that. It's called a slender loris. And some populations of slender loris in Sri Lanka and southern India only eat insects. I think it's harder to make a living that way, right? You're dependent on finding insects year-round. Insects can be a little bit seasonal. There are all sorts of strange things that go along with tarsier diets. Tarsiers and other haplorhines have lost the ability to synthesize vitamin C. I have no idea why you would lose that ability. Then you can get scurvy. That sounds great. Why not keep the ability to synthesize vitamin C? Anyway, it's a good question. I just don't know. How was that very long way of saying I don't know? Typical professor, right? Yes, hi.
My question is similar to the, baby lost call.
Yes.
But if we had a situation where, any of these leaping primates, they are carrying one of their children in their mouth, or they're in the farther along parts of the gestational period. How do they adapt to that significant shift in weight when they're moving so much weight over such a long period?
I love this question. I love it in part because nobody studied it and it's such a great idea. Somebody should be studying gravid, heavily pregnant tarsiers as they leap. Tarsiers are really understudied because they are extremely difficult to keep in captivity for any length of time. I only know of one place in the world that's a scientific facility that has live captive tarsiers that have had them for years. It's at an agricultural university in Bogor, Indonesia, south of Jakarta. And they somehow have been able to keep tarsiers alive. At the Duke Lemur Center, they had some tarsiers for a few years, but they just don't last long in captivity. And so it's hard to get to the places where you can study tarsiers. It takes a special type of person to want to get up at night and become nocturnal to follow your tiny little study animal around the forest. You have to worry about, so I've only seen tarsiers in the wild one time in central Borneo, so central Kalimantan province, and I was at an orangutan field station, and I was walking through the forest and I was thinking, hey, this feels pretty great. I got trails, and I looked down and I saw the giant snake about this thick and black headed down the trail in this direction. I never saw its head going by. And I said to the guy next to me, who was a Dayak guy from this area in Indonesia, and I said, hey, look at this snake. And I heard him go, oh, cobra. So, it's hard to study those sorts of things. So first you'd have to find primates, then you'd have to have fun to do it, then you'd have to wait for them to get pregnant and go and measure their leaves. I love it. So one of the cool things that my colleague, Liza Shapiro, here at UT, and her students did, a former student by the name of Catherine Whitcomb, she, for her dissertation, studied pregnant humans and how their center of mass shifts and how basically their lumbar spine repositions to accommodate the weight of a baby. It must be happening for tarsiers. Nobody's studied it. Okay, are you guys noticing a theme here? Everybody keeps asking questions. They're like, I don't know, but I'm going to talk for 5 minutes. Maybe they won't notice. Yes, hi.
Are some tarsiers poisonous?
No, I am not aware of any poisonous tarsiers. I'm only aware of one type of poisonous primate. It is an adorable, fuzzy primate from Southeast Asia. You find them in the same forest as some tarsiers. It's called a slow loris. And slow loris are said to have basically poison glands on their wrists. And what they'll do is they'll lick their wrists and they'll get these toxins in their mouth, and as they bite you, you can go into toxic shock. And they learned this, as I understand it, at a primate research center in Germany, when somebody got bit and they went into toxic shock. And then somebody else got bit a couple of months later and they went into toxic shock, and they were like, perhaps there is a connection. So anyway, yeah, so there's only one type of poisonous primate. It's not a tarsier, it's slow loris. Wouldn't that be amazing if they were poisonous?
Anybody from this side of the room, unrepresented in tonight's questions? Yes.
I was just wondering how you make money out of researching about smart bears.
So the question was, how do you make money out of researching tarsiers and fossils and things like that? The answer is, it's not easy. You have to be, this is the truth. You have to be extraordinarily lucky to land the sort of academic position that I have, or the sort of academic position that my wife Becca Lewis has, where, you're basically, because, last I heard, Facebook wasn't hiring primatologists or paleontologists, right? So it's one of the great features of academia in the United States, where you can do all of this really, really interesting, pure research that's not, admittedly, it won't cure cancer or make $1 billion, but at least for me, it's what I love to do, and I think it's really interesting. So in terms of funding your work, you're usually competing for grant money. And there are vanishingly small amounts of grant money to fund the sort of pregnant tarsier research we were talking about earlier. You'd have to convince an NSF review panel, like, I like that pregnant tarsier idea. Let's throw them a few $100,000. Did that answer your question?
Thanks for the talk. Really enjoyed it.
Thank you.
So, for a relatively small landmass, why are there so many different species of Tarsiers?
So, as I understand it, Dr. Banner could probably tell you more about this than me, but maybe he doesn't know about Southeast Asian tectonics. As I understand it, Sulawesi, not that long ago, was multiple independent islands that have only recently come together and sort of congealed, let's say. That's not the right term, is it? They haven't congealed.
You're doing fine.
Thank you. Geologists, close your ears. They basically formed one landmass. And so at least part of what you see in Sulawesi is that, as these landmasses have come together, these multiple species of tarsier have somehow managed to avoid competing with one another, maybe by occupying different types of forests, maybe by having slightly different ecologies. But I think that's part of the story as well, in addition to Sulawesi's isolation. I would say that our ability to understand tarsier species diversity is greater because of modern genetics than our ability to get into the wild and actually see what makes them tick, like different species. Like Tarsius tumpara, what's its ecology like? I don't think anybody knows, for the very reasons that we were just talking about, right? There are not millions of dollars out there funding tarsier research. And so most primatologists are doing this kind of work on a shoestring and making do and getting to the field when they can. The other thing I'll say, though, the flip side of that is, you maybe noticed that it's the same species of tarsier in Borneo as in southern Sumatra. 12,000 years ago, that was all one landmass when sea levels were low. So that helps to explain part of the story, but that's the opposite of your question. That's lack of diversity. Is that, you buying it? Forget I said congeal, please.
Okay.
Thank you.
Chris, we have a question here from the person who's driven the furthest tonight to get here, all the way from Sun City, Texas.
Well, actually from Dallas today, so we just got here. Yeah. Oh, 2 questions. That's terrific. Look at the video. The ears were moving a lot. Okay?
Yes.
And I was kind of interested to know what's going on there. And the second question is, do we know much about the size and the function of the cerebral cortex?
Oh, okay. So yes, and yes. The ears are highly mobile. They're basically parabolic sound collectors, just like your cat, right? Have you ever noticed that if your cat's looking away from you and you talk about your cat, you mention his name, the ears will rotate? They'll be like, what? Someone's talking about me behind my head, and they'll be listening. So if you have a parabolic collector that can concentrate sounds, as you sweep it across the space, let's say there's a sound source right here, you'll hear, right? As you sweep across it, you'll be preferentially amplifying once it's pointing in the direction of the sound source, particularly for the range of frequencies that size ear receives. So tarsiers are like a lot of other insectivorous primates in having mobile ears that are really good at localizing sound sources. So even in darkness, right, tarsiers will listen and they'll hear rustling in the leaf litter and they'll orient and they'll be looking, where is it? As far as the brain is concerned, tarsiers have got really interesting brains. One of the talks that I was just on at the same Society of Vertebrate Paleontology meeting is with my old advisor at Duke, Dr. Rich Kay. And Rich Kay presented on brains in haplorhines. And it turns out that tarsiers are like monkeys, apes, and humans in having a large proportion of their brain that is in front of the brain, particularly the cerebral cortex, this big wrinkly thinky part of your brain. In front of the central sulcus, it's the frontal lobe. Frontal lobes are big, but the back part of the brain is even bigger because that's where visual information is being processed. And so it looks like haplorhines have got these characteristic, let's call them cerebral cortex shapes that are also related to the fact that they've got high visual acuity and they're doing a lot of interesting things with vision. You take your typical monkey like a macaque and you start parceling up its cerebral cortex and you say, what percentage of the cerebral cortex deals with visual information? It's almost half. It's a lot. Did that answer your question?
We'll take one more question and then we'll see who wins the skull of the tarsier. You bet.
Hi. Oh, Jesus. Okay.
So you were talking about how the tarsiers are 100% carnivorous. I was wondering how that affected the actual insect population. Like, how did they adapt to combat with these mighty predators?
Please continue asking these sorts of questions. I don't have an answer for you. It's such a great question. I mean, so anywhere you see predators and prey that have been in place for a long time, certainly there have been insects in places like Borneo since the Eocene. There have probably been tarsiers in places like Borneo since the Eocene. You see these co-evolutionary relationships sort of, back and forth between predator and prey, just like, cheetah and gazelle on African plains evolving longer and skinnier legs so that they can both run quickly. You see, I can think of a few examples of interesting counter strategies that insects that are heavily predated by one type of insectivorous mammal will evolve. So for example, there are certain types of moths that have evolved little eardrums on their abdomens, and when bats are echolocating and basically closing in for the kill, they fold their wings and go into a spiral to try to evade them. So those sorts of adaptations do evolve. I'm not aware of anybody having demonstrated something like that for tarsiers. It's yet, I mean, listen, one of the things I love about being a scientist is you scratch the surface on any topic and you realize we don't know anything about anything. And so everybody here, you've already heard like 5 or 6 different dissertation ideas. And as long as they could get funded, right, they would be really, really interesting questions to ask. So it's a good one. I don't know the answer.
Awesome. Thank you, Chris. We now need you to think of a number between 1 and 320. And whoever has that ticket is going to get one of these really cool, personally signed by the speaker, Tarsier pictures.
183.
183. All right. It's a love letter from your husband. Well, let's try another number.
72.
72.
Yay!
I told you. All right. Give us another number and we'll have another sign.
146.
146. 146. Yeah. Come on down. And while they're coming down, how about another number?
What? It's between what?
1 and 320.
320.
320. Here you go. Enjoy it.
Anyone? They're not
320. Last in, first out.
here. How about 302?
302. Come on down.
302.
Go. Chin. Doing. Here, I'll take it in. Take it. You riff it in half. So if he says 20, you're ready, or if he says 3, you're also ready. Okay. All right. Next, believe it or not, we have.
Oh, nice.
It's a plushie. It's a plushie of a tarsier. What's the Venn diagram of plushies, all the plushies in the world, and all the tarsiers in the world, and they actually meet? Give us
88.
88. 88 wins a tarsier plushie. This is the cutest thing I've seen all week. I'm not sure I want to give this up. Yeah, okay.
188.
188.
Yay!
Yes. Here, remember. It's in your phone. No, it's okay. Keep it. It's a memento. Keep it in. Awesome. We have the last tarsier plushie.
11.
11. 11.
Another one
11.
back. Am I being random enough? I don't know if I've repeated anything. More lower numbers?
More lower number.
Like what?
Do you have your 11? Thank you.
Higher?
Ooh, what's next? Oh, man. Tarsier coloring book. You haven't really have a meditative, artistic, creative experience until you've colored in a tarsier.
Rainbow Tarsier, 132.
132.
All
Yes.
right.
These have been certified as correct.
Anatomically accurate. Oh, I saw a rhinarium. Hold on.
Another Tarsier coloring book. I thought that was the only one in the world.
Number 10.
10. 10, 10, 10. Yay! Why, this young man was coloring at the tables outside. Yeah, you're welcome. He is a natural colorer. That's awesome. In the Extremely Weird Animals book series,
Nice.
the Tarsier.
Nice. I hope that's volume 1. The pressure. 199.
199.
That is the correct range for Tarsiers. Very good.
So it's Congratulations. Thank you. Oh my gosh. Look at this shit. What? Can you?
Yeah,
Would you mind?
of course.
Look at that. What are you looking at? Wow. Angelina, where did you find this?
She noticed they gave him friendly primate teeth instead of scary teeth.
All right.
222.
222. All right.
Hooray! You'll grow into it. Here we go.
It shrinks when you watch it.
Yeah, right. Hot water.
Oh, what should we do first? Let's do this. Let's do the stickers. A Tarsier sticker. Look, it's got a human dental bridge.
It does, yes. Another smiling Tarsier.
Need another number.
Number 3.
3. Very good. Watch your step.
What am I leaving out?
We have 2 from the same family. There'll be an investigation.
Another one? What? It's between what? 1 and
320.
I think 305.
Feel fine. Yes. Usually there's more people who have left. They're really sticking around for the Tarsier swag. Okay, Chris, whenever you're ready, we'll do another one.
Okay, 169.
169.
That's big. They great.
169. Going once, going twice, another number.
139.
139. Whoa,
Yes.
that was a great reaction. Here, Chris.
Okay. He is this only for good, never evil.
We have another Tarsier sticker. Look, it's got
it's the
no
chunky
split up
Tarsier.
the left.
Wow.
It's got the giant man hands.
Okay.
Yeah,
it's
good
true.
there,
It's
yeah.
true. Well, that goes to lucky 111.
111. 111.
Yay!
Wow.
Okay. Thank you.
One more number, Chris.
219.
219. 219. Hey! Dr. Musgrove.
A distinguished geologist this week.
I hold in my hand the final sticker, but not the final prize.
Wow. It goes to 68.
68, Karnak.
I meant to say it goes to 71.
71, congratulations. Okay, let's get serious.
We weren't serious.
Yes, it's about everything that's highlighted.
Oh my gosh. The speaker has the option of keeping it.
Okay, so
He's not going to say a number.
well, gosh, this is really. It's got to be. 88.
88. You already said
Did I
88.
say 88 already? Oh, wow. 200? 214.
214. Wow. Yeah.
Remember, you're going to only eat bugs while you wear this, no matter when you do.
All right, that was a small. This is a medium.
Oh, well, that's going to go to 267.
267. 267? Yeah, yeah.
Woo!
I think
You
we should
definitely
have,
grow into
we
this.
should have a picture. They're going to take it.
Oh.
Please say here for a picture. Picture. Okay, maybe not. Yeah, you want to take a picture with the speaker? Okay. So kind of hold it up and like put it over your head and stuff like that.
And the other end of the dome, you know, you feel better.
Yes.
I hold in my hand the final giveaway.
This is the big one.
Look at this. John works there.
Anatomically accurate.
Softballs.
So this goes to number 200.
200. 200.
Maybe it goes to number 248.
248. Oh, so close. They got it.
You got it? All right.
Really good.
I think that's it.
Dr. Kirk will be happy to stick around and answer some questions, but why don't we let him know how we feel about what he did here tonight?